Okay, Assalamualaikum warahmatullahi wabarakatuh. A very good day to everyone. So we are going to continue on our topic from Neurobiochemistry. So this is the part 2 of the topic. Okay, so we are going to discuss on the process of impulse, of nerve impulse conduction along a neuron. Okay, we are going to highlight key components involved in the generation and propagation of an action potential. Okay, so we can see the structure of the nerve body, which is it has the cell body and dendrite. So this what we call is this soma. Okay, so the function of dendrites, as we know, that it going to receive the incoming signals, okay, which is neurotransmitters from other neurons or sensory stimuli, okay, and these signals cause membrane depolarization. What does it mean by membrane depolarization? So it is the condition where the resting membrane potential becomes less negative due to the influx of positively charged ions. Okay, for example, for the sodium ions. Then from there we go to the trigger zone. The trigger zone is located at the junction, okay, of the axon. Okay, this is the site where the summation, okay, of the graded potential, okay, from the dendrite and the soma occurs. Okay, if the depolarization reaches a threshold level, so an action potential is initiated. Okay, so if the threshold is reached, the action potential is fired. If not, no impulse is conducted. Okay, so the action potential propagates, okay, along the axon, okay, traveling as a wave of depolarization. Okay, so this propagation is facilitated by the opening and closing of voltage-gated ion channels along the axon's membrane. Okay, in the myelinated axon, okay, the action potential jumps between nodes of Ranvier through a process called saltatory conduction. Okay, so it helps in making the transmission faster. So we have this axon terminal, so the impulse reaches the axon terminal, where it triggers the release of neurotransmitters into the synaptic cleft. So what are the key concepts that we need to know about the process here is what does it mean by depolarization, okay, action potential, and also propagation. Okay, so this diagram, okay, explain, okay, about the process of action potential, generation and propagation in a neuron. Okay, so it focuses on the changes in membrane potential and ion channel activity. Okay, let's look at the resting membrane potential, which is at minus 70 MV. Okay, at this rest condition, the inside of the neuron is negatively charged relative to the outside. Okay, so this is maintained by the sodium-potassium pump, okay, which is sodium-potassium ATPase pump, which pumps three sodium ions out of the cell and 2 potassium ions into the cell, okay, by the relative permeability of the membrane, okay, which is the membrane is more permeable to potassium than sodium. Then we have this threshold of excitation, Okay, when there is a stimulus, stimuli, so it can cause the membrane potential to depolarize to a critical threshold. Okay, typically around minus 55 MV. So this condition will cause the voltage gated sodium channels to open and sodium begins to rush into the cell. Okay, now let's go to the depolarization phase. Okay, so this is where the sodium influx occurs. So once the voltage gated sodium channels open rapidly, so it allows sodium ions to enter the cell. Okay, to enter the cell. So the membrane potential becomes positive, rising quickly towards 40 MV. So this rapid depolarization forms the rising phase of the action potential. So let's look at the peak, of action potential, which is at the 40 MV. So at the peak of the depolarization, sodium channels becomes inactivated or become refractory, preventing further sodium entry. So now voltage gated potassium channels open, initiating the repolarization phase. Then we enter the repolarization phase of potassium. So now potassium efflux, where the potassium ions exit the cells through the voltage gated potassium channel. So this causes the membrane to move back toward the resting level, becoming more negative. So last but not least, on hyperpolarization. So as potassium channels close more slowly than sodium channels, there is an excessive efflux of potassium, so causing the membrane potential to dip below the resting levels, around minus 80 MV, because of the excessive efflux of potassium. So this phase basically ensures that the neuron does not immediately fire another action potential. So it allows for a refractory period. Okay, so after hyperpolarization, the sodium-potassium pump and leak channels restore the resting membrane, restores the resting membrane potential at minus 70 MV. So this extra potassium diffuses away and the neuron is ready to fire another action potential. So basically this slide summarize of what happened at the image in the previous slide. So you just need to know about what happened during the resting membrane potential, on the depolarization start, repolarization, hyperpolarization, restoration, and also action potential. Okay, now let's go to the synapse. What is synapse? So synapse is the junction between two neurons or between a neuron and also the target cell. So it is composed of presynaptic terminal, synaptic cleft, and also postsynaptic membrane. So what is the function of the synapse? One is for the neurotransmitter release. So when an action potential reaches the axon terminal of the presynaptic neuron, so it triggers the release of neurotransmitter. Then these chemical messengers are released into the synaptic cleft from vesicles into the presynaptic terminal. So once a neurotransmitter is released, so this neurotransmitter will start to diffuse the synaptic cleft and bind to specific receptors on the postsynaptic membrane. So this binding can either excite depolarize or inhibit which is hyperpolarize the postsynaptic neuron depending on the type of neural neurotransmitter and receptor involved. And last but not least, there is a process of reuptake and removal. So because not all neurotransmitters are absorbed by the postsynaptic neuron. Okay, so unused neurotransmitters are often reabsorbed. Okay, unused neurotransmitters are often reabsorbed by the presynaptic neuron through a process called reuptake. Okay, so now let's look at the communication at the synapse. Okay, so the ratio of synapse to neurons in human for brain is 40,000 to 1. So you can imagine how fascinating the process of communication that occurs at the synapse. So it also indicates the high level of connectivity and complexity in neural networks. Okay, so we know that the function of neurotransmitters, because it plays a very critical role in facilitating communication between cells at the synapse. So it also you can see from the image, it shows an intricate structure of neurons and also synapse, highlighting their connectivity and interaction in the nervous system. Okay, look at so now let's move to the components of synapse. Okay, so there are 3 components of synapse. We have presynaptic terminal, synaptic cleft, and also postsynaptic alignment. So what do we have in the presynaptic terminal? So it consists of vesicles that filled with neurotransmitters synthesized by the neuron. Okay, and for the synaptic cleft, this is basically a gap between presynaptic and also postsynaptic neurons. Okay, so this is where neurotransmitters is released, and we have postsynaptic element which contains receptor that binds neurotransmitter, leading to signal transmission. So what is the process for the neurotransmitter? Okay, so the neurotransmitter is synthesized in the presynaptic neuron. So they are packaged into vesicles and released into the synaptic cleft. Okay, when vesicle fused with the presynaptic terminal, presynaptic membrane. So neurotransmitters then bind to the receptor of the postsynaptic membrane. So it activates the postsynaptic membrane. So the excess neurotransmitter are either metabolized, transported back into presynaptic terminal, or it will diffuse away. So there are 2 types of synapse. Okay, one is excitatory synapse, which is type 1, and we have type 2 synapse, which are inhibitory synapse. So basically, more to discuss about synapse. One, what is the difference between excitatory and also inhibitory synapse? Then what is GABA synapse? This GABA synapse is inhibitory. So it have less postsynaptic thickening. So it is basically inhibitory in nature for GABA synapse. So we also have glutamate synapse. This is an example of excitatory synapse. So it features more postsynaptic thickening and contain more vesicles. So there are 7 types of synapse in together. Alright. So this is about the images that shows different types of synaptic connection. We have dendritic, exodendritic, extracellular, exosomatic, exosynaptic, exonic, and also exosecretory. Okay, so what happened in dendrodendritic? So this is where the communication occurs between dendrites and dendrites of two neurons. So dendrites going to send message to other dendrites. However, exodendritic, the axon terminal of one neuron synapse of the dendritic spine of another neuron. So this is a common in excitatory synapse. Okay, so extracellular. So axon terminal is going to release neurotransmitter into the extracellular fluid without a specific target. For exosomatic, so this is often associated with inhibitory signal where the axon terminal synapse directly on the cell body of another neuron. Okay, how about the exosynaptic? So axon terminal synapse on another terminal, okay, influencing its activity. We have axonic, okay, the axon terminal synapse on another axon. For exosecretory, the axon terminal connects to the blood vessel, releasing neurotransmitter directly into the bloodstream. Okay, now let's move to the chemical synapse. Okay, we have discussed on the component of synapse, okay, which consists of the synaptic cleft, and also postsynaptic membrane. So how does neurotransmitter release, okay, by the action potentials in presynaptic terminal? Okay. When an action potential reaches the presynaptic terminal, it triggers the opening of voltage-gated calcium channel. Okay, so calcium ions enter the presynaptic. Okay, it enter the presynaptic terminal. Okay, so it triggers the synaptic vesicle to fuse with the membrane and release neurotransmitters into the synaptic cleft via exocytosis. Okay, then neurotransmitter travel, okay, across the synaptic cleft to bind with specific receptors on the postsynaptic membrane. Then neurotransmitter will diffuse. Then the binding of neurotransmitter to receptor, okay, is going to activate the postsynaptic element. So it triggers the opening of the channels to open. So sodium ion enters the postsynaptic cell, depolarizing the membrane. If the depolarization reaches the threshold, it triggers an action potential in the postsynaptic neuron. Okay, so this is the events at a chemical synapse. Okay, starting from the arrival of nerve impulse, okay, calcium influx, okay, then the release of neurotransmitter, calcium removal, diffusion across synaptic cleft, receptor activation, and also termination of neurotransmitter action. So basically it consists three events, okay, at chemical synapse. So try to understand what happened, okay, at every single events. Okay, what happened during the arrival, what happened to the calcium influx, and also termination of neurotransmitter event. So on the electrical synapse, okay, so let's look at the structure of the electrical synapse. So we can see that electrical synapse, it consists of the gap junction, okay, that allow the direct flow of local current between cells. Okay, this is the gap junction. Okay, so the function of this electrical synapse, basically the action. So gap. Okay, let's discuss on the gap junction. So it allows local current to flow between adjacent cells. So what is connexons? So connexons are protein tubes in cell membrane. Okay, so this is a protein channels that form this gap junction. Okay, this is the connexons that form this gap junction. Okay, so the action potential of one cell can directly triggers an action potential in the neighboring cell. So this allows a rapid and synchronized communication between cells, okay, functioning as if the connected cells were a single unit. And these electrical synapses can be found in cardiac muscle and many types of smooth muscle. So this is where the electrical synapse, okay, located. Alright, so the importance of this electrical synapse, okay, it is critical in tissues that require synchronized contractile activity such as heart, okay, to ensure efficient pumping of blood. Okay, so it's a bit about neurotransmitter. Okay, so we have discussed on the structure of neurotransmitter and all that. So let's discuss on the 5 conditions, okay, that it must be met before we call something as a neurotransmitter. 1, it must be present in terminal. Okay, because we know that this substance must be synthesized and stored in presynaptic terminal. So it should be readily in the synaptic cleft. Okay, synaptic vesicles to be released when the neuron is activated. Okay, release on fire rate. So this is another requirement, okay, of condition, okay, for the neurotransmitter. Okay, the substance must be released, okay, into the synaptic cleft when an action potential reaches the presynaptic terminal. So this release typically occurs through the process of exocytosis triggered by the calcium influx. Then placing substance emulates firing. What does it mean? Okay, when the substance is artificially applied to the postsynaptic membrane, it should produce the same effect as when it is released naturally by the presynaptic neuron. Then uptake for inactivation. So the mechanism must exist to inactivate or clear the neurotransmitter after its release. Okay, and also the last one is activation block stimulation. So blocking the neurotransmitter's action or uptake mechanism should prevent further stimulation of the postsynaptic cell. So this can occur through drugs or toxins that inhibit receptor binding, reuptake or degradation enzyme. Okay, these are the 7 steps in neurotransmitter transmission. Okay, we have synthesis, storage, release, receptor interaction, inactivation, reuptake, and also degradation. Okay, so synthesis, we know that neurotransmitters are synthesized in the presynaptic neuron, either in the cell body or axon terminal. Okay, for storage, neurotransmitters are stored in synaptic vesicles within the presynaptic terminal until it's needed. Then release upon arrival of an action potential, calcium influx triggers the fusion of synaptic vesicles with the presynaptic membrane, releasing neurotransmitters into the synaptic cleft. Then we have receptor interaction. Neurotransmitters bind to specific receptors on the postsynaptic membrane, initiating a response, which is the opening in ion channels or triggering intracellular signaling pathway. Then we have inactivation. The neurotransmitter's action is stopped, okay, preventing continuous stimulation of the postsynaptic neuron. Then we have a reuptake. Okay, the unused neurotransmitter is going to be reabsorbed into the presynaptic neuron or nearby glial cells for reuse. Then we have, last but not least, we have degradation. Neurotransmitters are broken down by enzyme in the synaptic cleft, ensuring signal transmission.
Malam.
Hai.
Tadi Ami kata tadi malam tadi awak mas.
Macam mana? Tak faham.
Amir kata Mak Lang tak datang rumah Aiden dengan Muaz.
Abang Amir kata?
Marah dah dia.
Aduh, Abang Amir ni kan.
Mak Lang kat ofis dia tak rumah Mak Lang.
Mak Lang dah kat rumah. Mak Lang tengah buat kelas ni.
Oh jangan deh. Mak Lang tengah work lah.
Bukan. Mak Lang tengah buat recorded je.
Okey.
Oh, malam nanti main dekat.
Alamak.
Aku malam nak.
Alah, Mak Lang tak tahu lagi. Nanti Mak Lang okey, Mak Lang datang ya. Tak minggu ni, minggu depan ya. Dah sedih dah pula. Dah terbenung pula.
Nanti banyak lagi kan?
Cuti? Ya.
Ini cuti lah.
Kita cuti banyak ke sikit?
Banyak.
Banyak. Nanti cuti banyak. Nanti Mak Lang boleh dah datang kan?
Makan sekarang.
Bukan.
Lah.
Bukan. Mak Lang work. Nampak ni Mak Lang work. Mak Lang work. Aiden kenapa? Aiden bangun awal sangat ni.
Ini cuti lah.
Alah, Mak Lang work.
Mak Lang work.
Mak Lang work ni. Mak Lang work ni.
CD rosak ke
Aiden.
Bye.
Adik. Dah dapat ke? Dah check ke?
Lah, tak dapat lah. Dulu tak dapat.
Tak apa. Ada rezeki je. Tak apa. Okey je. Apa?
Dekat je rayuan.
Buat je rayuan. Dia kan SPP ni hari tu last kali intake bulan 12. Kau tahu tak?
Kak Ina tahu lah. Budak ofis Kak Ina anak ni masuk bulan 11.
Betul?
Tak tahulah dia orang. Aisyah dah cuti dah tak dapat.
Tak apa. InsyaAllah. Okey. Rayuan je. SPP sepanjang tahun. Aiden nak ikut Mak Lang pergi work ke?
Nak.
Boy. Kenapa jadi gala biru? Apa?
Okey.
Macam.
Bapa Mak Lang lah. Bapa Mak Lang tu.
Mulut. Dah macam-macam. Terror lah Aiden. Terror sangat ni.
Mak Lang nak work. Bapa Mak Lang.
Kenapa Aiden rambut Aiden merah? Biden. Dia jadi hantu ke? Dia dah jumpa dah. Biden. See you Aiden. Aiden makan apa? Bye. Bye Aiden. Aiden nak tengok Mak Lang ke ni?
Dah lah tu.
Aiden makan apa? Aiden makan apa, Den? Aiden makan apa?
Macam apa dah?
Tak tahu.
Bye. Mana letak tau? Bye. See you. See you Aiden. Mak Lang ada lampu Aiden. Mak Lang ada lampu. Aiden nampak tak? Lampu Mak Lang ni.
Astaghfirullahalazim.
Banyak.
Mak Lang ada lampu. Aiden nak lampu ke? Mak Lang ada lampu bulat. Nak ke? Aduh, terbenung. Tangan tongkat pipi tu kenapa? Aiden call Maksud tu. Aiden call Maksud cepat. Maksud datang rumah Aiden kata. Cepat. Okay, so this slides basically summarize on the neurotransmitter family. Okay, so we have this neurotransmitter can be divided into small molecule transmitter, neuropeptides, and also gaseous neurotransmitter. So what is small molecule transmitter? So it can be it can be divided into 4 component: acetylcholine, monoamines, amino acid neurotransmitter, and also ATP and its byproduct. So what is acetylcholine? So acetylcholine is very important for muscle activation and autonomic nervous system function. For monoamine, so it can be further branch into catecholamines and indolamines. So basically it involved in mood regulation. Okay, for catecholamine, sleep wake cycles, and also circadian rhythms. And for indolamine, so we have serotonin and melatonin. This one is for mood regulation. Okay, so basically it's similar, almost similar to the catecholamines. For amino acid neurotransmitter, we have glutamate, we have GABA, and other amino acids. Okay, so basically this is glutamate is the main excitatory neurotransmitter in the central nervous system, and GABA, this is the main inhibitory neurotransmitter in the CNS. Okay, for ATP and its byproduct, so it acts as a neurotransmitter or neuromodulators in some system. For neuropeptides, okay, so it includes larger chain-like molecules such as endogenous opiates, okay, substance, hormones, okay, cholecytokinin, insulin, vasopressin, oxytocin, and more than 40 others neuropeptide. And we have for gases neurotransmitter, we have nitric oxide and also carbon monoxide. Okay, so basically nitric oxide is involved in vasodilation and memory formation. Okay, and carbon monoxide is a potential neurotransmitter with less established roles. Okay, receptor. So what is the importance of receptor that we need to know? So the function, okay, the significance role of receptor. We know that neurotransmitter binds to specific receptors on the postsynaptic membrane, initiating a response. So it resembles this lock and key model. Okay, the binding, okay, to the specific receptor by using lock and key model. So basically where the neurotransmitters as a key fits into receptor, okay, as the lock at the specific binding site. Okay, and it has this ion channel. So many receptors are associated with ion channels that open and close, okay, when activated. Okay, for the drug interaction, okay, receptors are an important target for drugs, okay, as they can be activated, blocked, or modulated to influence neural activity and treat various conditions. Okay, let's look at the receptor molecules and neurotransmitters. As we know, one of the important characteristic is between receptor and neurotransmitter. It is very specific. Okay, a neurotransmitter only fits into one receptor, okay, similar to a lock and key mechanism. And one thing that we need to know now, not all cells have receptors for every neurotransmitter, so making communications is more targeted and also specific. And And it has dual roles because neurotransmitters can have different effects, either excitatory effects or inhibitory effects. So when we talk about excitatory effects, okay, either it can promote or promoting on action potential, for inhibitory, suppressing an action potential. Go for presynaptic and postsynaptic action. So some neurotransmitters like norepinephrine bind to receptors on both postsynaptic and also presynaptic terminals. So the binding of norepinephrine to postsynaptic neurons basically is to delay the signals for the binding to the presynaptic terminals, basically to inhibit further release of neurotransmitter. Okay, so for the removal of neurotransmitters from the synaptic cleft. Okay, so this is the method. Okay, the methods on the removal of neurotransmitter depends on the neurotransmitter itself and also the synapse. Okay, so acetylcholine. So how does acetylcholine involve in the removal of neurotransmitter? One is by this enzyme acetylcholine esterase. Okay, breaks down acetylcholine in the synaptic cleft. So it produces acetic acid and also choline. So this choline, okay, choline will be recycled and transported back into the presynaptic neuron, where it's going to combine with acetyl-CoA to synthesize new acetylcholine. Another one, we have norepinephrine mechanism. So norepinephrine is taken back into the presynaptic neuron. So this is the reuptake mechanism. And also some epinephrine can diffuse away from the synaptic cleft. Okay. And also enzyme norepinephrine, enzyme monoamine oxidase, okay, inactivates norepinephrine prior to be absorbed into circulation and is going to be broken down into the liver. So here a summary of the key points regarding acetylcholine removal. Okay, removal of transmitter from synaptic cleft. Okay, by acetylcholine mechanism or norepinephrine mechanism. Okay, based on the explanation that we have discussed in the previous slide. Okay, so for acetylcholine, this is another detailed discussion on how acetylcholine involved in removal and recycling of neurotransmitters. So we know that this enzyme, acetylcholine esterase, degrades acetylcholine in synaptic cleft. So it will broken down the acetylcholine into choline and also acetate. So choline, okay, is going to be reuptake, okay, the transport protein and also V-type proton ATPase, okay, is going to return the acetylcholine to vesicles. So this transport protein basically help in recycling choline back into the presynaptic terminal. Okay, then we have the resynthesis of acetylcholine. So within the presynaptic terminal, cholinesterase combines choline with acetyl-CoA to resynthesize acetylcholine. Then endosynaptic formation of synaptic vesicles. Alright, endosynaptic formation of synaptic vesicles. Synaptic vesicles are reformed through endocytosis, allowing the neuron to prepare for another round of neurotransmitter release. So we have neurotransmitter. Okay, more discussion on neurotransmitter. So in definition, we know that neurotransmitter are chemical messengers that facilitate communication between neurons and between neurons and the other cells. They are essential for transmitting signals within the nervous system and between the nervous system and the rest of the body. So what are the key features of the neurotransmitters? So there are over 50 different neurotransmitters that have been identified, each with unique roles and also effects. So neurotransmitter can be categorized into two primary ways. One is chemically classification and functional classification. Based on their chemical structures, acetylcholine, biogenic amines, and peptides are the examples of neurotransmitters. So we know that acetylcholine plays a key role in motor control, memory, autonomic nervous system. For biogenic amines include neurotransmitters like dopamine, serotonin, norepinephrine, and histamine, which are crucial for mood, and other functions. For peptides, such as endorphins and substance which are involved in pain modulation. For functional classification, neurotransmitter can be classified by excitatory or inhibitory, and also direct nomotropic or indirect metabotropic, based on the ability to open gate channels. So we know that neurotransmitter plays diverse role depending on the type and classification. So we can summarize as it has a very important function in regulating mood, which handled by serotonin, dopamine, controlling motor movement, modulating pain, and also influencing memory and learning. So here's a brief explanation about chemical neurotransmitter. Acetylcholine. I think we have discussed that. I think you can add more information about how these chemical neurotransmitters, the examples, what is the, for example, biogenic amines, it includes dopamine, serotonin, norepinephrine, histamine. So add more information, add more notes, on every single point, of this chemical neurotransmitter. Same goes to this excitatory and also inhibitory neurotransmitter. What is the role of acetylcholine, aspartate, dopamine, histamine, or epinephrine, for this neurotransmitter? So I think you can summarize, on these notes on acetylcholine. What happened? How does acetylcholine plays their role in a neurotransmitter? So we know that acetylcholine is the first neurotransmitter identified and is one of the best understood. So it has been released at neuromuscular junction, where it stimulates muscle contraction. Also, it also can be released by neurons in autonomic nervous system. So this acetylcholine synthesized within neuron and start in synaptic vesicles for release. For degradation, it broken down in the synaptic cleft by the enzyme acetylcholine esterase into choline and acetate. In terms of the receptor binding, acetylcholine binds to cholinergic receptors, which are classified into two types, which is nicotinic receptor and also muscarinic receptor. So for nicotinic receptor, it can be found in muscle, and also certain autonomic neuron for muscarinic receptor can be found in parasympathetic target organs. So let's look on the acetylcholine synthesis. So acetylcholine is synthesized in cholinergic neurons, acetylcholine is formed from two components, from choline and also acetyl-CoA. So choline is obtained from the diet or recycled from the previous neurotransmitter breakdown. So acetylcholine, we know that is produced in mitochondria as part of cellular metabolism. So enzyme, the reaction is catalyzed by this choline acetyltransferase. This enzyme transfers an acetyl group from acetyl-CoA to choline, forming acetylcholine. So this acetylcholine, once synthesized, is going to be stored in synaptic vesicles within the presynaptic neuron, okay, ready for release upon an action potential. So for cholinergic synapse, okay, this is more on information about what is cholinergic synapse. So synaptic vesicles, synaptic vesicles in the synaptic knobs store acetylcholine with approximately 10,000 molecules per vesicle. So you can imagine how significant, okay, is this acetylcholine, okay, and also the synaptic vesicles. So when an action potential arrives at the presynaptic membrane, okay, it depolarizes the membrane, causing calcium channels to open. So it will cause the influx of calcium, and we're going to trigger vesicle to fuse with the plasma membrane. Then acetylcholine is released into the synaptic cleft and diffused to bind with receptors on the postsynaptic membrane, initiating a response in the cell. Okay, what are the toxin effects? Okay, so I'm sure you have heard about botulinum toxin. So this is one of the toxin can inhibit acetylcholine release, leading to muscle paralysis. Okay, another toxin is laterotoxin from the black widow spider. So it's going to overstimulate acetylcholine release, causing excessive neurotransmitter. Alright, so this is the diagram illustrating acetylcholine release and the action potential. Try to understand what happened at the resting state during the arrival of action potential, what happened during the acetylcholine release, and also during the receptor activation. Okay, I leave it to you to summarize, okay, the diagram on the acetylcholine release and also the action at the synapse. Okay, so this is one slide on the botulinum toxin, okay, Botox. Okay, so it's a neurotoxic protein produced by the bacterium Clostridium botulinum and related species. So in terms of the application, it has been commercially produced for medical, cosmetic and research purposes. Okay, for medical use, it includes treating conditions like muscles spasms, chronic migraines and excessive sweating. For cosmetic, we know that it has been used to help to reduce the appearance of wrinkles by temporarily reducing facial muscles. Okay, in terms of the mechanism, okay, this botulinum blocks the release of acetylcholine at the neuromuscular junction, leading to temporary muscle paralysis. Okay, in terms of the effectiveness, okay, when a person has been injected with a small amount of this toxin, it weakens the targeted muscle for 3 to 4 months, providing temporary relief or aesthetic improvement. Okay, so you can see the before and after use of this toxin, so it can help to smooth the wrinkles around the eyes and other facial areas. Okay, a little bit on the enzyme. Okay, acetylcholine esterase enzyme, which hydrolyzes the neurotransmitter acetylcholine. So, and it has performed its function, okay, in the synaptic cleft. So it has two active sites or two subsites, okay, both which are important to the breakdown of acetylcholine. One is an ionic site which binds the acetylcholine molecule to the enzyme, and we have another one is esteritic subsite, okay, which catalyzes the hydrolytic reaction. So the ester bond of acetylcholine is cleaved at the esteritic subsite. Okay, then this reaction produces acetate and choline is going to be taken up by the presynaptic neurons, okay, via a high affinity cholinergic system. So this is a diagram of acetylcholine hydrolysis by acetylcholine esterase. Okay, look at how the binding of choline, okay, where the acetylcholine binding which at the anionic site of the enzyme through electrostatic attraction, and this position acetylcholine for hydrolysis. Then hydrolysis occurs at the esteritic site. Okay. So this is another slide for the okay, this electrostatic attraction. Okay, and this is explain how the acetylcholine hydrolysis by acetylcholine esterase. Okay, the binding at ionic site and the hydrolysis at the esteric site. Okay, this is another diagram that illustrates, okay, on the acetylcholine synthesis, release and breakdown. Okay, so synthesis, cholinesterase, acetyltransferase. Okay, synthesize the choline by combining choline and also acetyl-CoA. Okay, by combining choline and also acetyl-CoA. Okay, then acetylcholine is stored in synaptic vesicles. Okay, via a vesicular choline transporter within the presynaptic neuron. Okay, then upon stimulation, acetylcholine is released into the synaptic cleft. Alright, then acetylcholine binds to postsynaptic receptor, okay, initiating a response in the postsynaptic neuron. Then this acetylcholine will be broken down in the synaptic cleft, breaking into choline and also acetic acid. Alright, and the choline transporter reacted choline into the presynaptic neuron. How about cholinergic receptor? Let's look at the cholinergic receptor. Okay, so this nicotinic receptor can be found at neuromuscular junction of skeletal muscle. So all gangliogenic neurons in the autonomic nervous system. So this, we know that nicotinic receptor basically is an excitatory neurotransmitter that trigger action potentials or muscle contractions. Another one is muscarinic receptor, can be found at parasympathetic target organs, for example cardiac and osmosis matter. So it's generally is excitatory receptor, but its effects can vary depending on the organ, also receptor subtype. So in general, basically these are the functions of acetylcholine. Okay, what are the functions of acetylcholine in the body, and also what is also the importance of acetylcholine in medical field. So we know that brain cholinergic neurons are critical in Alzheimer's disease, Huntington's chorea, and also generation of epileptic seizures. Okay, and this one is another point of biogenic amines. Okay, this is the elaborated explanation on biogenic amines. So we know that the amines has two types, catechol amine and also indole amines. In terms of distribution, these biogenic amines are widely distributed in the brain, where they act as a neurotransmitter that influence neural communication. Okay, function basically more on the emotional behaviors and also on the biological clock, on the circadian rhythms, okay, sleep-wake cycle, and also daily physiological process. So let's look on the synthesis of catecholamine. Okay, so this begins with the amino acid tyrosine, which serves as the parent compound. Okay, then we have the stepwise enzymatic pathway. It starts with the tyrosine hydroxylase that converts tyrosine into L-dopa. Then we have dopa decarboxylase that converts L-dopa into dopamine, and then we have dopamine beta hydroxylase that converts dopamine into norepinephrine. Then we have phenylethylamine and methyltransferase to convert norepinephrine into epinephrine. Okay, dopamine and norepinephrine are synthesized in the axonal terminals of neurons, while epinephrine is primarily synthesized and released by the adrenal medulla as a hormone. Okay, this is more detailed information about biogenic amines, especially on the norepinephrine and dopamine. Okay, how these two biogenic amines plays a very important role in regulating mood, motor function, and also autonomic nervous system activities. Okay, I mean you can just read this. Okay, so a bit on the serotonin. Okay, synthesis. So it can be synthesized from tryptophan. Okay, another amino acid. Okay, so the synthesis of serotonin involves two reactions, which is hydroxylation, which tryptophan is converted to 5-hydroxytryptophan by the enzyme tryptophan hydroxylase, and it requires cofactor, which is tetrahydrobioprotein, which is converted into dihydrobiopterin during the reaction. So the reaction is the first and critical step in serotonin production, which plays a very essential role in mood regulation, sleep, and other physiological function. And next step, a decarboxylation step, is the final stage in serotonin synthesis to produce serotonin. So serotonin is very, very important, okay, in regulating numerous physiological and physiological process, and its dysfunction is linked to conditions like depression and also anxiety. So this is about the breakdown of serotonin. So serotonin can be degraded into reaction. One is oxidation, which 5-hydroxytryptamine is oxidized by the enzyme monoamine oxidase, and this convert this serotonin into 5-hydroxyindoletriacetaldehyde using oxygen and water, followed by dehydrogenation, which this molecule is broken down by the enzyme aldehyde dehydrogenase to 5-hydroxyindoletriacetate. So this is the primary metabolite of serotonin. So this 5-HIAA is excreted in the urine and is often used as a clinical marker to evaluate serotonin metabolism. So these two-step degradation process ensures a proper regulation of serotonin levels in the body, preventing excessive neurotransmitter activity. So if you have any any questions related to the topic of neurobiochemistry, I think we can have a discussion during class, or we can have personal discussion as well. But I would strongly suggest and advise you to summarize, okay, in the mind map form to enhance your understanding. Okay, thank you very much.